Hello, folks. It's a concerned Dr. Miskoff on August 6th, 2020, long day at the hospital. Fortunately, not so many COVID patients, at least that I took care of, only two at this point in time that I personally saw, but, Perry 10 in the local hospitals that we round at or less. So the numbers continue to drop as far as hospitalizations, or at least stay stable at Perry 10. That's a good thing, although we're seeing all the other medical illnesses, as I've mentioned on previous logs, from respiratory failure, GI bleeds. Eventually, those patients are going to come in, DKA, etc. Tonight, I'd like to cover monoclonal antibodies. It's getting some press. Dr. Fauci and some of the other larger organizations and drug companies have some news in regards to monoclonal antibodies. We've talked about the immune system, we've talked about convalescent plasma, and now it's time to take a look at, cloned antibodies that are taken from original parent cells of patients who recovered from COVID-19. Of course, this would be utilized in patients who are either prophylactically, who are exposed to it, potentially in patients such as healthcare providers that are do not have COVID-19 but are going into high-risk situations or heavy hit populations. New York, New Jersey, obviously, were hit hard and early compared to the rest of the country that's seeing it now. So potentially as a prophylaxis, I had mentioned in earlier vlogs, if you go back, that I thought that when we saw vaccines, we would see it coupled with monoclonal antibodies. So potentially giving providers or high-risk populations, it could be nursing home populations, obviously, prisons, places where there's large groups of people hanging out on a consistent basis, could get monoclonal antibodies to protect them potentially from developing COVID-19. So that's the idea. Or treating patients who are outpatients and more mild, and hoping that they don't progress to more severe disease, or patients who are admitted with more moderate or severe disease and hopefully preventing them from progressing to overt respiratory failure, mechanical ventilators, ECMO, and then whether if they're at that level, studying them as well. This is basically direct competition to convalescent plasma. Convalescent plasma obviously being harvested from patients who have recovered, obviously pretty recently from COVID-19, at least when we first started giving it. Now I imagine there's a pool of a little bit older donors, but it can't be too old as it does have a shelf life. But this would be basically a synthetic form coming from the original recovery cells of these patients. So I believe they try and go back as far as they could to those original cases in Washington state that were reported. But there's so many companies, which is a good thing, working on these treatments. And as a general rule, if it ends in MAB, M-A-B, like boy, M like Mary, A like Apple, B like B, then it's typically a monoclonal antibody or an antibody. Monoclonal, or recombinant antibodies, meaning that they were cultured or grown out, the clones, not from a live mammal like a mouse or a rabbit or something like that, but more from a virus or a yeast. So when they talk about recombinant, that's what that means. And monoclonal, meaning it's basically binding to the one epitope on this antigen. Remember, an antibody is produced by the B cell, and then it recognizes that antigen, whether it be, again, a bacteria, fungi, or a virus, or a chemical, or whatever it is. If a antibody reacts to the patient's own body or own organs, then we have an autoimmune disorder. So you're attacking yourself, such as multiple sclerosis or some of the rheumatologic conditions, lupus and stuff like that. At any rate, what are human monoclonal antibodies? Again, they're proteins produced by the body's immune system in response to that antigen, that harmful substance, and it could be a infectious disease or some other foreign body. And this is just explaining when you have an autoimmune condition like rheumatoid or multiple sclerosis, then you're attacking your own body. Monovalent affinity to that one epitope, that's the term monoclonal. They can design ones that have more than one binding site on that antigen. But what basically is happening here is you are injecting these mammals, typically, again, if it's not recombinant, with the virus or the antigen. And from their cells, the spleen cells, they can merge them together or fuse them together with myeloma cells. Remember, myeloma, multiple myeloma, is a cancer. It is a B cell cancer, if you will. And remember, B cells in the immune system lectures we did earlier are the ones that produce immunoglobulins. We talked about T cell reactivity the other night and how that's a different part of the immune system with T helpers and cytotoxic Ts, natural killers in memory. So this will be a B cell tumor producing immunoglobulin ultimately, but fusing it with that injected mammal with the antigen. This hybridoma then getting put into culture with a whole process here, and what comes out, these harvested monoclonal antibodies from the original parent cells. So they can go back to those early patients and do a process like this, and there are newer processes obviously going on for making monoclonal antibodies. This is the original description, if you will. That idea of magic bullet was first by Paul Ehrlich, for a fun fact, and won a Nobel Prize. I like this last name, Miskoff. So they won the 1908 Nobel Prize in Physiology and Medicine for that work, talking about multiple myeloma and some other stuff there. As far as the clinical trials go, some news from the National Institute of Health. Clinical trial to test antibodies and other experimental therapeutics for mild and moderate COVID-19. Again, you'll see studies in those outpatients who have milder disease, trying to get them within a week or so. They'd have to have a positive test, going into phase 2 trials, and after that comes the phase 3. Everything's getting accelerated, type of Operation Warp Speed approach, if it's reasonable. And Eli Lilly is in charge of this in conjunction with Abcelera out of Vancouver. So they're working together, and there's many other companies working on this. So it'll be interesting to see how that works and how it's provided. So if you have the coronavirus with its spikes, the spike proteins coming out, that's obviously where the antigen is, and you're going to have these monoclonal antibodies attach to receptors and neutralize, destroy whatever it has to do. You do a search on clinicaltrials.gov, you come up with over 40 studies that are recruiting, some of them new, a lot of them are in China and other parts of Europe. You can see Regeneron's getting going with this, and they have different levels of studies, from ambulatory to hospitalized, sicker patients and more severe, to, I believe, more prophylaxis studies as well in development. Saw the Eli Lilly with the NIH there, and you got some out of University of Washington, Arizona, all different ones being looked at. Again, if it ends in MAB or MAB, tends to be a monoclonal antibody. There it is, MAB, monoclonal antibody. Lenzilimab, you can see all the different ones here listed. Some of them are just numeric, but a plethora of different monoclonals from big companies and smaller ones looking at that. And you can go on clinical trials, as I've shown before. This one got some press recently, the leronlimab, hard to say all these, they start to blend together, out of California. And that one is cytodyne, which also being looked at, I believe, in breast cancer and other. A lot of these are going to cross over with the hem-onc world, as we've seen with the crossover with rheumatology. Although disappointing on sarilumab and tocilizumab so far, there were some other trials listed on here. Remember, forms of different monoclonal antibodies. Some are against the viruses, and some are directed other ways. So I do think that this is going to be one of the most effective therapeutics potentially out of all that we've seen. In quick review, dexamethasone, only one shown to reduce mortality. How we dose it outpatient in the mild disease, who knows? And I don't see those studies really being done, although I'm sure somewhere, hopefully, we'll see different doses, because 6 milligrams for an inpatient that you can monitor makes sense. For an outpatient with milder disease, we don't know if that's the right dose. Is the nebulized way to go. Remdesivir, obviously shortening the course in those moderate severes. How about the mild? How about prophylaxis? Rumor on the street is we may see either a pill form or maybe even a nebulized form. A little bit upset about the concept of us, being directed to never use nebulizers initially, because of aerosolization, but some of these studies will look at different types of nebulized devices like mesh nebulizers, sort of tighter fitting mask nebulizers that rather than losing 20, 30, 40% of the drug, you're delivering closer to 100% of the drug, which therefore would reduce aerosolization potentially, hopefully. So, these are the therapeutics that are promising or already shown to be beneficial. Again, the anti-IL-6s, we haven't seen anything grossly positive and even some negative stuff. Stop trials already there. Convalescent plasma, we think we'll see some more positive stuff besides cases and anecdote. But again, will the monoclonal antibodies that are made the way we showed or similarly be able to be mass produced, and can it be provided to the masses is one of the questions. But if there's so many companies working on it, it is promising. And maybe they can all work together at some point if that's possible. And whether or not just one monoclonal antibody is enough, or do you have to hit, there's so many different spikes on the coronavirus. Are all antigens created equal, or on the proteins? So the question is will you have to give more than one monoclonal antibody at a time, or cycle them is still to be determined. Anyway, we'll end it with that. Again, many more therapeutics to continue to talk about in regards to COVID-19. Haven't really gotten into the vaccines too much. I find the monoclonal antibodies a little bit more interesting, but time will tell how it all pans out and how we give them, and together or separately, prophylactically with the monoclonals, or is it going to be more of a treatment type thing? Time will tell. Everybody have a great night, and we'll see you soon.